studying the bactericidal properties of Limulus polyphemus cell-free serum, or the blood from the American horseshoe crab with the clotting cells removed. This is a continuation from last year where I used the whole blood, or the hemolymph, of the horseshoe crab in order to test for inhibition against 2 different kinds of bacteria. So this year I decided to see whether there was some other defense mechanism in the horseshoe crab besides the clotting cells that are responsible for inhibition in the horseshoe crab. So I developed 4 different protocols in order to test my project. My first protocol was testing whole cell-free serum against the 3 bacteria that I used last year, which were Escherichia coli, Enterobacter aerogenus, and Staphylococcus epidermidis. My second protocol was testing whole versus boiled serum against the bacteria to see whether proteins were responsible for inhibition. My third protocol involved removing hemocyanin from the serum. And my fourth protocol was using a salting out method of ammonium sulfate precipitation to divide the proteins into 2 different protein solutions. So my basic methods were to inoculate a aliquot of bacterial solution, which was bacteria inoculated in autoclaved water, and add that to serum samples that I would be using between protocols. And after the allotted time, which was normally a nominal time 0 and anywhere between 30 to 120 minutes, I would place that on nutrient agar and incubate that for 24 hours in an incubator and count the colonies. So for my first protocol, I was using whole cell-free serum against E. coli, Enterobacter aerogenus, and Staphylococcus epidermidis. And as you can see with E. coli, between 0 and 120 minutes, there was a significant reduction in the number of bacterial colonies. However, with Enterobacter aerogenus, very little inhibition was observed, and with Staphylococcus epidermidis, there was an increase in the number of colonies. So I could conclude that there was most potency against E. coli, so I would use E. coli for the remainder of my experiment. For my second protocol, I used whole and boiled serum against E. coli, and as you can see with the whole cell-free serum between 0 and 120 minutes, there was a significant reduction in the number of colonies. However, with the boiled serum, there was not that much significant reduction. Therefore, I found that the proteins in the serum were responsible for the inhibition of bacteria. For my third protocol, I used centrifugation in order to remove the hemocyanin, which is the major protein found in the serum. And I tested the whole cell-free serum, the hemocyanin-reduced serum, and I also resuspended the hemocyanin and tested that against the bacteria. And at nominal time 0 compared to water control, the whole cell-free serum and the hemocyanin-reduced serum had the greatest reduction. And even after 120 minutes, the number of colonies further decreased with both those samples. Therefore, I found that I could successfully remove the hemocyanin and still maintain inhibition of bacteria. And in my fourth protocol, I used ammonium sulfate precipitation, which is a salting out method, in order to divide the proteins. And so I had a 50% supernatant, which were proteins dissolved in the solution, and a 50% pellet, which were proteins not dissolved in the solution. And at nominal time 0 compared to water control, the whole cell-free serum and the 50% pellet had the greatest reduction, and after 120 minutes, the number of bacteria further decreased. Therefore, I found that the ammonium sulfate pellet had the greatest inhibition of bacteria. So future advancements will be to hopefully use column chromatography, which is a more specific procedure in order to fractionate the proteins to find which proteins have the greatest